Okay, so we're leaving the nerves. But before we get there, how did the ice feel when it heard it was fired? Broke? It was crushed. Wah, wah, wah. Let's see, what does a cow say backwards? What does a cow say backwards? Very good. Nicely done. You got to make a fool of yourself if you can participate. All right. How about this one? What does a peanut say when it sneezes? What is a peanut? Peanut. Catch that second part? What does a peanut say when it sneezes? You have to enunciate. Kachoo! Well, I thought it was fun. You guys are getting more difficult as the semester winds down. I'm telling you. You're harder to entertain. Alright. I had colored pencils. I had 3 jokes. I have what else to do. Yeah.
Are you going to open up a quiz and worksheet for this?
I am. In fact, the quiz and the worksheet was opened moments ago. So the quiz, let's do the worksheet. The worksheet is due next Monday, a week from today, by 11:59 p.m. That's April 13th. The quiz is going to be due Tuesday, the 14th, by 11:59 p.m. Now, you guys know the drill. It will submit automatically. You just need to save your work. You don't have to hit submit, and you can do it ahead of time. Okay? Now, a worksheet and a quiz coming up means part of the semester is training you all. So I've trained you to expect what? When's the exam? What's that? Next Wednesday. Somebody say bonus. Okay, good. Next Wednesday, okay? We have 3 lectures in muscle. And the importance of our format is strategic. If you notice, we're actually in the textbook going backwards. You guys see that? Because we were in nerve physiology, which was material that we are supposed to cover after muscle. If you look at the chapter numbers, and if you look at the organization of the textbook, and many textbooks are this way. They put muscle physiology first, and then they put the nerve physiology. I actually like to do it the opposite. And the reason for it is just to be rebellious, I guess, but I think it makes sense. It's now in the semester that hopefully we have some common understanding of how an action potential works and what it means. We have some common understanding of what triggers it. What does it do? What determines if you fire an action potential versus just a local or a graded potential? When an action potential or an electrical signal that crosses threshold, known as an action potential, gets to a target cell or tissue or organ, like a skeletal muscle, it's going to trigger a response. And most of the tissue in the body isn't going to do anything all by itself without nerve innervation. Okay, there's sort of a rare example, and the only example is the heart. But then you would argue that it is autorhythmic. It doesn't need nerve innervation to tell it to beat because it's contractile all on its own. Because it has its own neuronal circuitry that is independent of the nervous system. Right? You're going to learn about that in 202. You're going to learn about the electrical pathway of the heart. Starts with, you don't need to write this down. It's just part of the soapbox introduction, so we just kind of slide into it instead of abrupt. So there's an SA node, and it has an auto-rhythmic beat of about 100 beats a minute. And it sends its electrical signal throughout the Purkinje network, throughout the heart. And histologically, those intercalated discs that you looked at in the beginning of the semester, we said it electrically couples one cell to the other. What that means is when one cell fires, it knocks on the door of this adjacent cell and says, it's your turn. And so then they go in sequence. Now, you can have innervation onto the heart to extrinsically control it, but if you take the heart completely and de-innervate it, it'll beat all by itself at about 100 beats a minute. And that's what happens with heart transplant patients. With heart transplant patients, when we get them, we do not reconnect the neuronal circuitry. We'll usually put a pacemaker in, and then you can dial in the rate. But if you put a heart transplant from one patient into another, number one is you have to immunosuppress the recipient. Number two is when you get the donor, you only have about 4 to 6 hours to actually do something with it. And of course, it's in cardioplegia cold solution to keep the tissue preserved. And they air vac it from one location to another, so it's only within about a 4 to 6 hour flight pattern that you can actually do this. You immunosuppress the donor, the recipient. Donor doesn't care that it passed. And you put a pacemaker in. If you didn't do that, you would run at about 100 beats a minute. So every target tissue that we see is going to have electrical impulses going to it. Or it's going to, if it's a sensory cell, it's going to have electrical signals coming from it. In lab this week, you're finishing out your last new material. We're just a unit behind. But it's nerve physiology again. Right? You're talking about special senses. You're talking about gustation. You're talking about olfaction. You're talking about vision. You're talking about hearing and equilibrium and balance. And all of these special senses, they're located in the head, all use electrical impulses to send the signal. Sends the signal not very far because it's already located in the head to the brain, and then you interpret it. And then it sends a motor output to something to say, it's coming from over there. And you follow your nose. Okay?
Dumb question. Does that mean if you have a heart transplant and you experience a fear or something, your heart rate won't just increase? You don't just stay even until you get...
No, that's not true. Because the heart itself is going to be perfused with blood flow. And in the blood, you're going to have adrenaline hormone. So it eventually will make it to the heart and bind to those receptors and cause the heart rate to go up. I'm talking about the basal rate. It's the vagus nerve. The vagus nerve is what slows the heart rate down from 100 to resting. So you don't reattach the vagus nerve, which is the parasympathetic decelerator, if you will. Okay? Wait till next semester. We'll get a lot more detail. So this is our introduction to muscle. Why do we do nervous tissue first? Well, we do nervous tissue first because the muscles are the response organ, but they have to respond to a signal, and that signal is an electrical impulse. So the muscles themselves are a significant component of our body mass. Right? They make up nearly half of our total weight. There's over 600 skeletal muscles in the human body. Most oftentimes they're found in pairs because we're bilateral organisms. But not always. You can think of some examples like the ubicularis oris or the anus. Now, these muscles that line the GI tract are not going to be found in pairs because it's just a single tract. But when we look at like the biceps brachii, there's two. And you remember back in laboratory, you'd have to discriminate between right versus left. Okay? The muscles that are skeletal in nature, we're going to talk towards the end of this unit about cardiac and smooth. But the bulk of our discussion is on skeletal muscle. That's sort of our main model. And we're going to set it up with skeletal muscle. We're going to go to all the details about skeletal muscle. And then we're going to use that platform to say how it's similar to cardiac and how it's different than smooth. And that's the rest of the material. So it's voluntary skeletal muscle. So again, for the next 1.5 to 2 lectures, we're going to spend mostly on skeletal. This means voluntary requires conscious stimulation. And the functions of these muscles are listed here. Now, some of them are pretty obvious, like movement, going from one place to another. Stability, maintaining posture. You're using skeletal muscles right now to sit upright. Control of body openings and passages, like around the mouth, the avicularis oris, the avicularis oculi around the eye. The GI tract, the urethra, the anus, for the elimination of waste. Skeletal muscle also helps to generate heat. It produces about 85 or as high as 85% of our body heat. And that heat is important not to just keep you comfortable. It's not just about being comfortable. One of the reasons you're comfortable at a certain temperature is that your homeostatic set point and enzyme function is optimized. Remember, in the very beginning of the semester, we flash all the way back to our first and second lectures when we were together. Right, the weather was different. You were wearing a yellow dress. I'm just kidding. Ew, weird. Why, you don't like yellow? No, that was the weird part? If we flash back to the beginning of the semester, we talked about some of those early lectures on metabolism and homeostasis. Homeostasis is about keeping things optimized. And what do we know that's true about enzyme function if you raise the temperature too much? It denatures the protein and it doesn't work anymore. Okay? So this body metabolism and this body homeostatic set point, temperature-wise, is really important. If we look at skeletal muscles and how they facilitate movement, you don't have movement unless the muscle crosses over a joint that bends. You know, you can't bend your arm if you flex your bicep muscle. You can't bend it right here. There's no joint. So muscles have to cross over a joint, and the way that they work is they shorten. They contract, like if you have a puppet. Has anybody ever used a puppet, seen a puppet, Googled and heard about a puppet? Okay, sweet. That should accomplish everybody. So these puppets, if you tug on the string, then the puppet moves because there's a bending joint at that location, and we don't operate too differently. We're a lot more sophisticated, obviously, and we don't need a puppet master necessarily, but we have to cross a joint in order for movement to be facilitated. So therefore, skeletal muscles have two attachment sites. They have to attach above the joint, they have to attach below, superiorly and inferiorly. And the origin is defined as the point of attachment that doesn't move. It's the non-moving part. Some of you are like, we had to memorize the sublist of origin, insertion, and action. I could never remember what the origin really... The origin doesn't move. Where it inserts is actually where it moves from. But it's relative. Like, so for example, if I say that I'm going to flex the thigh, right here, then this is the origin and this is the insertion. Okay? But I could also say that I'm going to flex the thigh and I can flip origin insertion, because I could do this. Okay? Now that example doesn't exist for every single muscle. So not every, some of you are like, oh, I got a test question I'm going to challenge. That's one of the few examples that it works. It doesn't necessarily work for like the biceps brachii, because the biceps brachii, it only goes in one direction, right? You don't sit here like this and then, you know, flex the rest of it up. Okay? Is that, you guys with me? So the hip flexor is like a convenient example to, you know, for some of you to go, oh, I knew I didn't miss that practical quiz question. My TA is so wonderful. So each muscle, though, has to have at least one action. And many times they have multiple actions, because if you imagine, like the puppet example of a string coming across, our muscles are like a bundle of string, and they don't necessarily come in a straight direction. They can actually come at an oblique angle across a joint. So when you pull or contract, you can take that joint, especially if that joint is not a single plane moving joint. If it can move in more dimensions and you pull at an oblique angle, you can get a flexion and a twist at the same time. Does that make sense? So the complexity isn't just in the muscle and how the muscle wraps around the joint. there's complexity in the joint itself. And that's why when you studied in lab, the different types of joints and the ones that were more movable, and the ones like the ball and sockets, and the ones that were less movable, like a hinge joint. A hinge joint only moves in one plane. In the ball and socket, like our shoulder, it moves in multiple planes. And so we have many muscles of the rotator cuff that insert at the level of the shoulder, and when you contract them in concert, you can get these complex motions like throwing a ball, okay, waving at a friend, grabbing a backpack off of the back seat, okay? The muscle contraction typically moves the insertion closer to the origin, because the origin's not moving. And the innervations are the nerves. Like in lab, we have you skip a lot of the innervations, right? Or all of them, essentially. The volume of material is so much. But I want to make sure that you understand, so I'm taking the time to say it, these muscles that you're learning about won't do anything if there's not a nerve that tells it to contract. So the innervation, even though we don't have you memorize it in the laboratory, it's not trivial. It's actually very, very important. So if you cut those nerves or you lose those nerves, you cannot control that muscle anymore. And so we'll talk about deinnervation in the next lecture. What happens when you deinnervate a muscle? Well, number one is it atrophies or it shrinks, but we'll talk about some other types of things that happen. And there are toxins that do this and poisons and things. Okay? If we look at some of the crossover between lab and lecture, we see some terms that are familiar, like prime mover. Prime mover is the muscle that's responsible for the particular movement. The main muscle, the primary muscle. Synergists are muscles that assist that prime mover. They coordinate the same type of action. They help facilitate the same movement. An antagonist opposes that prime mover. So they're going to have opposite activities. Our example, biceps brachii, is our prime mover. It causes what? Elbow flexion, okay, or flexion at the elbow. The synergist is the brachialis. It assists that muscle in its activity. And then our antagonist is our three-headed muscle, right? Triceps. The triceps brachii. Okay, so that's a little bit of review and introduction. Trying to link some of the early concepts from the beginning of the semester. Trying to link some things out of lab. Trying to explain why it may appear that we're out of order with the textbook. Okay, and then the last thing that I'll say is, why is it that we're not completely in sync with the lab? Well, it's not possible. The amount of physiology that we cover in a lecture is so much more than the time that you have over the certain sections or systems in lab. So they don't necessarily completely line up. They, at times, come together and link up, and they're going to be complementary, but they're not completely parallel. All right? So hopefully you're tracking, because you've covered muscles pretty extensively in lab. But our interest in lecture is less about memorizing the muscles. Now you should have some competency. When I show a muscle, like the biceps brachii, and I talk about origins and insertions, and I talk about motion and hip flexors, we should be on the same page. And that's the intent. Now, if I want to get deeper than that, and I want to start diving into the microarchitecture of muscle, this is where lab and lecture are very different. In lab, you looked at the microscope and you looked at pictures of the muscle, but you never really got the story about what's happening at that level. Right? You didn't get the story about what makes it striated. Why does it look striated? How does it contract? What's happening? How do we regulate that? Why is it important? If we look at this muscle belly, this is our femoral head. You can see our tendon coming out. The Golgi tend to be located at that location. You can see this outer wrapping of this deep fascia. But I want to dive down to the level of the muscle fiber. The individual muscle fiber is here. Now that individual muscle fiber is a muscle cell. Just like when we looked at a similar type of picture with a nerve, we looked at a nerve fiber. That was a nerve cell. So like a nerve, that's a specialized cell. You guys agree with that, right? It's just a cell. It's intimidating because it looks different. It's got a big old fat body with a soma, and then it has like these projections that bring in signals known as dendrites, and then it collects the impulses at the axon hillock. It sends it down this long, skinny part of the cell, which is called the axon, and then it moves out from there into its axon terminals and into a button or a bouton where it's going to release synaptic vesicles that contain acetylcholine, and now we're at that synaptic cleft, right? But all that stuff in between is just the inside of a cell. Now I'm going to show you another specialized cell. I also like to do it this way because I think it's easier to make the connection that an axon and dendrites and the soma of a neuron is just different parts, bless you, of a cell. This is also a specialized cell, but there's some moving parts now. So now we're going to get even a little bit more complex than we did at the level of the neuron. If we look at this cell, it's multinucleated. Right? Our neurons were uninucleated. If you take a number of these muscle cells or fibers and you bundle them together, you surround them with a perimysium. Around the individual cell, you have this endomysium. I should have said that first. Endomysium. You put a number of these cells together and you wrap them with a perimysium, perimeter. You get a number of these vesicles, just like we had with the nerves, and you put them together, and you surround those guys with this epimysium, like epidermis, right? Like this outermost covering. So this architecture should look very familiar. It should look like what we saw with bone and with nerve. And this cross-sectional view, now that I have my pencils, is really simple to see. When you take a bundle, and this was the example I was trying to show last time, but I didn't have all these pencils with me. And we just take one of these rubber bands, and you wrap it around. Okay? And those in the back can't see this, but you can kind of imagine what it looks like. Right? So that's this view. That's a cross-sectional view as you're cutting through. And each of the individual pencils is maybe one muscle cell and one muscle fiber. Okay? So we're going to get down into the individual muscle fiber. In lab, you spent all your time out here. We're going to spend most of our time actually inside the cell. So very different than where you were. So characteristics of muscle cells, and I'll get to your question. Number one is they're excitable. Excitable means that they have the ability to receive and respond to a stimulus. The response is actually to contract. And so they're going to do something if you trigger them. They're responsive, or they're excitable. Number two, conductivity. They have the ability to produce an electrical signal down the entire length of the muscle. So they can conduct that signal out or propagate it. It's not enough if the individual muscle says, oh, I know what to do with that signal. You want it to actually send the signal to the rest of the muscle cells. Contractility. That means they can actually shorten. They're contractible. Extensibility. Not only can they shorten, but you can extend them back to their residual or resting length. And we'll talk about structures or features that allow them to do that. They give muscles their elastic recoil. I mean, the last thing you want to do is, I've got an itch, I scratch it, and it's like, oh crap, going to be stuck here for a little while. Okay? Could you pull my hand down for me because it's not extensible. It's only contractible. Okay, so that would be strength throughout our day, especially if you're trying to walk, because it's a series of motions of contracting and relaxing, contracting and relaxing. They're elastic. Muscles are extremely elastic. They have an ability to recoil. So what you don't want to do is that you need to scratch your temple. You're like, oh man, I can bring it back, but it takes a while. I'm going to push the next slide here in a second, but I have to wait for it because it's not very elastic. So you want your cells, when they're going to go back to their extended state, you want them to go fast. You want them to go arguably just as fast, if possible, as when you contract it. Okay? It'd be a very awkward running gait if you could contract quickly, but the relaxation or extensible side was very slow. Okay? Question up here.
The muscle fascicle, does that term refer to the bundle of muscle fibers and also the bundle of bundles?
The muscle fascicle is a group of muscle fibers that are in a pod, if you will, wrapped with an endoneurium.
Okay.
That's the fascicle. Okay? There's a question over here.
So someone with faster reflexes, does that just mean like their muscles are more elastic, or?
It's a good question. Keep going. Or what?
No, that's just.
Or, end of question, not question mark. Okay. There's just like an or, and then, okay. So reflexes. Reflexes actually change quite a bit as you age. And they get slower. Reflexes are in their prime in your teenage years. Okay, actually all of you from a reflex standpoint are probably, it was better last year or the year before. Okay? So in your teens is when the reflexes are at their prime. And I'm not talking like, I'm still 19. Yeah, you're on the downward slope. Okay? Like I'm talking like 17. 17's probably the peak. Okay? So the reflex is a combination, to answer your question, of a number of parts of the circuitry. So one of the main parts is that sensory neuron that's bringing information in. The second main part is that inner neuron that's actually sending it back out to the motor neuron that goes to the muscle. And so those neuronal pathways are optimized in our teenage years. And then the signals, the rate at which signals flow slightly decreases as you get older. Then the muscle fiber question. There's a couple of different examples of muscle fiber typing that we'll talk about later in the unit. We'll talk about slow twitch, we'll talk about fast twitch, and then we'll talk about an intermediate type of fiber. And that composition can change. Then the other thing is the relative composition of titin can change from one individual to another. So titin is this elastomeric recoil protein that has it come back. And so my titin is probably different than yours, which is different than yours, which is different than yours. They're probably different isoforms. And so that also has something to do with who has faster circuitry with the optimized titin protein. And that's why you see that not everybody can be a professional athlete. But you only see professional athletes really rise up in the younger period of time. That's why we start talking about people that hit their 30s as a professional athlete as they're starting to wind things down. Right? Because all of those optimized things are behind them. Now they're probably playing with training and experience and understanding the game, but eventually the physique wins out when the younger and younger players keep showing up. Okay, a couple other questions and then...
I was going to ask, what's that endomysium? Sorry,
the endomysium?
Yeah.
The endomysium is this outer covering that goes around an individual muscle fiber. The endoneurium goes around the individual nerve. Remember? So, yeah, that's a great question. Endoneurium was the counterpart in the nerve picture. You guys remember that? That's it? There wasn't another hand? All right, I'll move on. I know if I wait long enough, somebody will ask some question. Okay, so now let's dive in. So there's a lot of questions. This is the most confusing part that I lose everybody. So let's back up. What does that look like to you? Pencil. Okay? Muscle fiber, someone can read. Great. Sorry, I didn't mean to read. Sorry, I totally threw you under the bus like there. So I publicly apologize. It wasn't meant to come out that, smart aleck. Yes, you are correct. Very good answer. Thank you for participating. What else? What else does it look like? I will try to refrain from being such a jerk. Pencil. Muscle fiber. And then it went downhill from there. Fascicles. Looks like fascicles. Okay, let me back up. If you're familiar with this is all familiar, right? And we dive down to this level, okay? The individual muscle fiber. This is the individual muscle fiber. See it circled, the yellow part? So that is one of those. So we're diving down into the individual muscle fiber, and now you're looking inside the cell. So this is a cell, one cell. Nobody said that. This is a multinucleated cell, because here's a nucleus, here's a nucleus, there's a nucleus, okay? This is the inside of the cell, and this is this membrane, which we call the sarcolemma on a muscle fiber. The sarcolemma. And inside of the cell is the fluid, what we would call the cytoplasm in a regular cell. Here we actually call it the sarcoplasm. And you can't see it because it's clear, but you can see where the picture is pointing is it's pointing to the inside of the cell where there would be cytoplasm if it was a typical cell. But because it's a specialized cell, we call it sarcoplasm. Okay, you guys following? But what about all this junk, Keller? What is all that stuff? Well, these, if you blow these up, these are myofibrils. And if you look down at the inside of the myofibril, these are myofilaments. And these myofilaments are primarily actin and myosin, and they're proteins. And actin and myosin exist in all cells. They're the cytoskeleton architecture that's inside cells. You guys remember cytoskeleton from intro bio? And the cytoskeleton allows the cell to change its shape. Typically, we talk about in introductory biology when the cell is going to divide. Remember when the cell was going to divide, and then you had to look at an onion slice? You guys remember that? You're looking at an onion slice. You don't remember that? Wow, you missed out. So the onion slice is pretty cool. You can still do this on your own. But if you looked at the onion slice, you're looking for different phases of mitosis. You guys remember that? Prophase, interphase. Remember that? So when the cell is going to pull apart, it's made another copy of its DNA, and it's going to separate out. It's the cytoskeleton that allows it to actually pull apart. Okay? So every cell has actin and myosin. However, in a muscle cell, it's very specifically organized. That's the difference. And you're seeing this very uniform architecture, which is kind of cool. It's very strange, but it's quite cool. And if you look at, you see this blue, it looks like, some sort of web-like work. See that? This sort of blue pattern. So the artist is actually just terminating it here, and you can see how it ends. But this architecture is contiguous with a couple of things. So for example, you probably can't really see it on the drawing, but if you look in your textbook, you're going to see a hole. So it's like the surface of the membrane, and then you have like a manhole, like in a street. And the manhole covers off. And the reason for that is you want to be able to send an electrical signal down the membrane, and you want it to be able to go deep into the muscle itself, the muscle cell, and interact with these myofilaments that are in the middle of the cell. And if they're in the middle of the cell and the electrical signal's out here, how do they get to the middle? Well, they have to come down one of these, what we call T-tubules. So the T-tubule is this purple tube that's diving deep into the muscle cell, into the middle of the cell. Because you remember on nerve physiology I told you, that action potential, where is that action potential causing a difference in electrical signal? Where in the cell? All throughout? Only at the surface? Deep within the middle of the cell? It's actually only at that surface. Do you remember that? So it's just beneath the surface is where you're changing ions. Now you propagate it down throughout the length of the cell, but if you move into the middle of the neuron, it's electrically neutral. Okay? So the architecture here allows, with this transverse tubule, we call it the T-tubule because that stands for transverse, and it makes a T. As it comes here, it comes down. As it comes here, it comes down. And so every so many distances, you're going to get a T-tubule diving into the inside of the cell to allow that electrical signal to go into the middle of the cell so that the middle of the cell stuff can respond, not just the stuff at the surface. Okay? We'll come back to how does it respond. Right now we're just looking at the architecture. But like I've said all semester, the way the anatomy is set up should give you some insight as to what the physiology is. So you can see that this cell is very different than any cell you've ever seen. Is that true? Yeah. So that must mean that it has some specialized function, and it absolutely does. Now, the blue that's sitting next to the purple, this blue here is part of what we call the sarcoplasmic reticulum. The sarcoplasmic reticulum, it's abbreviated to SR. So that's all these small little networks right here. You can see sarcoplasmic reticulum, SR. And at the very edges of this sarcoplasmic reticulum, there's kind of like a bulge. Do you see how these little blue small ones lead to this larger blue tube that's kind of bulged out? That's called a cistern, which means tank. And it's called the terminal cistern, or terminal cisternae, which is the end tank. Because it's at the end of the sarcoplasmic reticulum on both sides. Do you guys see that? So the terminal cisternae, the big blue bulges or big blue tubes, are contiguous or connected, that's what that word means, connected to the sarcoplasmic reticulum. What does the sarcoplasmic reticulum do? It stores calcium. We'll come back to that later. But right now it's important for you to understand that the sarcoplasmic reticulum, or the SR, stores calcium and makes it readily available here in the future when we talk about what it's going to be needed for.
So a muscle fiber is one muscle cell, correct?
One muscle fiber is one muscle cell, correct.
So the T2 carries electrical signal across on the perimeter and periodically dies down to the inner side?
No. No, the sarcolemma is the membrane. Remember how in a neuron, the electrical signal just cruises across the membrane, unless it's myelinated? If it was myelinated, then it dove inside and then popped out. So that's what happens here. Just like in a nerve, the electrical signal is going to come down the membrane. The membrane here is called the sarcolemma. When it hits the T-tubule is when it dives in. So it cruises across until it hits the hole and then it goes down. Okay?
So through a whole bunch of red stuff? So it gets
Yes. So it gets all throughout the entire cell. Essentially at the same time. Okay, so we're going to see this diagram. We'll talk about this A band, I band, and Z disc here in a moment. But the other thing that I want to show you is, you see these mitochondria? These are all mitochondria. What do mitochondria do? Powerhouse of the cell. They make ATP. You're like, yeah, these are all these little buzzwords we learned in intro bio. Yeah, well, that's why you do that. Right? Some of you were so frustrated with some of these introductory classes, but I promise you, we do it this way so that when you get to this point, we can actually make reference to a cell And you know, you don't look at us funny, like, oh, I don't want to sell those. What's that? Okay. Mito what? Okay. ATP what? So the mitochondria generate ATP, and you drew cells in intro bio that had one nucleus and had one mitochondria, right? But this cell has many nuclei, and look at it's packed full of mitochondria. So what does that tell you about the energy demands of a muscle? Extremely high. Very high. Okay? So a lot of these are connecting the dots of stuff that you already know. I get the benefit of telling you why you learned about all that stuff. If you're looking in the future and you're going to be practicing medicine and you're going to be looking at treating patients, and one of the side effects is a mitochondrial enzyme toxin, right? Adverse events is it poisons mitochondria. Do you think that's a problem? If the patient takes too much of it? Yeah, they'll suffocate, right? You'll paralyze their respiratory muscles. So, why? Who cares about mitochondria? That was intro bio. Well, yeah, because all that stuff runs everything that you are learning about. Okay? So we get a lot of these comments that are like, I'm so bored because we covered this and covered that. And I just want to like start slicing up people. You know, I'll be a surgeon. Say, well, you know, slow down, Turbo. I mean, we got to sort of do this incrementally. And this is why this stuff's important. And there's so much info that we have to layer it for you. We have to layer it so that you can actually absorb and digest it over time. That's why it takes a couple of years. Okay, so these Z-discs, I-band and A-band, these are structures that describe different locations relative to actin and myosin, relative to the myofilament. So we're going to cover those in a lot more detail in two-dimensional pictures because they're easier to understand. But I wanted to point them out to you. When you feel like you understand the 2D picture, go back to the 3D picture here to see if you can wrap your head around, all right, what's moving, what's getting shorter, what's happening. You with me? Okay, so let's go to a 2D picture. This is a simple 2D picture. But if you look at this picture, and you see the A band here, and you see the I band, and then I back up, here's the A band, and here's the I band. Do you see this A band's kind of got like this purple region? And then the I band doesn't have the purple region. Do you see that? And the I band has like the zigzag Z-disc that goes down the middle of it. Well, that's exactly what we have here. We've got our zigzag Z-disc, otherwise known as Z-line, that goes down right here. There's one right over here. We've got our purple A band, and we've got our sort of nothing zone from our 3D picture. It didn't look like there was anything there. There was just like this black band going down the middle. There's stuff there if you see. We're going to talk about those structures. But this I band is this little gap right here, which is this gap right here. Okay? Everybody with me? So let's back up, because I want to zoom out. Where are we? Here's our muscle and lab. Here's our muscle and lab. Here's our femoral head. We zoom in. Here's our little box. We zoom in here to a bunch of these muscle fascicles all bundled together. We dive down to muscle fascicle made up of individual muscle cells, otherwise known as a muscle fiber. If I zoom in on this fiber, I get to this picture. If I zoom in on this right here, right, I get to this picture. Did you guys follow the zooms? Just like Google Earth, like, right? Okay. So now we're at this level. This is the cartoon. This is an electron micrograph. And the cartoon is lined up with the different regions on the electron micrograph. You see how this like shading kind of zooms in to show you kind of where we are? So let's focus on the cartoon first, and then you can go to the electron micrograph. Well, we've got this purple segment here, which I called the A band, right? These muscles have this overlapping pattern of this A band and this I band. So it has this sort of thicker region or this darker region, and you can see the darker region right here. And then it has this lighter region where the thicker protein structure doesn't exist. And so this lighter region is called the I band. So when you look at this microscopically over an entire muscle, you see this striated pattern of these alternating A bands and I bands. And that's what you visualized under the microscope. You were looking at this, you just didn't know it. Okay? Now, if we start naming some of these zones, we're going to talk about the different structure details. For example, start at the bottom. A sarcomere. A sarcomere is referred to as the functional unit of muscle. A sarcomere is the functional unit of muscle. And it is defined architecturally from a Z disc, or the Z line, to the Z disc or Z line. So from here to here. Okay? Now, the artist draws it like a true Z. If you look at it in nature, this is an electron micrograph. The reason it's portrayed as a Z is you can see that this line doesn't sit right on top of this one. So there's your little Z. And then it kind of zigzags down and zigzags down. You see that? So that's that Z interpretation that we get. So from here to here is one functional unit of muscle. Now what's inside the function? Well, we have what we call a thick filament. The thick filament is this purple, larger protein. And then we have a thin filament. The thin filament is this very thin, that's why it's labeled thin, like orangish red globular protein. And we'll talk about what they're made up of later. But for right now, let's just call them thick and thin. Make sense? Then we have a third filament known as our elastic filament, which is this green one. Now I'm going to jump ahead and I'm going to cheat. I'm going to tell you the whole punchline. And then we're going to fill in all the details. So if you imagine, take your fingers like this, you imagine them interacting like this. Okay, everybody do this. Okay? If you want to lengthen the muscle, you're going to slide them this way. If you want to contract and shorten the muscle, you're going to slide them overlapping each other. Make sense? Well, when they contract and they move like this, in order to get them back quickly to resting, that elastic filament is like a rubber band. So that elastic filament allows it to snap back quickly. This elastic filament is this little green one down at the bottom. Okay? The elastic filament is made up of a protein known as titin. The elastic filament is made up of a protein known as titin. It's an elastomeric protein. We actually have one of the world's experts on titin physiology right here at NAU. Okay, Dr. Kisa Nishikawa. So her lab is doing active research on the titin protein and is actually kind of redefining the textbook on how this is organized. So when all of you are here, you're probably going to be using slightly different pictures after she has proven the way it works in a slightly new, twisted way. Just a new explanation of a model that we've been studying for a long time. Okay. You can see a couple of other features like the H band. So the H band is this area that only contains the thick filament. And it has no thin filament component. The M line runs right down the middle of the thick filaments, and the thick filaments are anchored in place by that M line. We defined the sarcomere as the Z disc or the Z line to Z disc or Z line, and that anchors the thin filament. It also anchors the titin protein.
The M line does all that?
No. The M line only anchors the thick filament. The Z disc anchors the thin filament and the titin protein. So these actin and myosin proteins, they occur in all cells. They function typically in the motility of the cell. They aid in mitosis, like we talked about, and the transport of intracellular material. But here they're organized in this very, very unique pattern. And as if we look at this pattern, and we look at some pseudo-colored electromicrographs, you can actually appreciate the striating pattern. So here we've actually pseudo-colored the fiber itself in this blue. Okay? So this is the native picture. Here it's the fiber that's colored this blue. Over on the right are the individual nuclei are colored blue. So you can see here's a nucleus, here's one, and that's what's showing up here. Down here we've highlighted in this pseudo-blue the A band. And then over here we've highlighted the I band. And so you can see these alternating patterns give rise to this very unique architecture of muscle. Oops, sorry. There we go. Okay. So the striations. If we look at these striations, the A band stands for anisotropic. Anisotropic. Now that's a property of being directionally dependent. So it's organized in a dependent fashion based upon the direction of where the fibers go. The A band is where the thick and the thin filaments overlap, and it's especially dark because of that. The H band, we already said, was thick filaments only. The M line is the middle of the H band, where the thick filaments are anchored. The I band is this alternating lighter band, and it stands for isotropic. Isotropy is a word that implies that it's identical in all directions. So it's not directionally dependent. And the reason that it looks a little lighter is because it reflects polarized light differently. So it gives it a lighter view when you're looking at it with light microscopy. And then lastly, we said the Z disc or the Z line, right? Borders the entire, or defines the entire sarcomere, and provides this anchorage site for thin filaments and elastic filaments. Now, if we look at some more pictures, we can kind of define these features based upon these images. So here's our myofibril. Here's a sarcomere that's going from Z disc to Z disc. You can see below it, colored in the pseudo-blue, the Z discs. You can see here the I bands. Down the middle is the M line, and on either side is the anchored thick filament. Where the thick filament exists only is this H band, and the A band is where they overlap, right? Thick and thin. So, lastly, we'll define this before we end. Our sarcomere, Z disc to Z disc, functional contractile unit of muscle. I kind of already let the cat out of the bag of how this works, how these overlap and slide. But they shorten because the individual sarcomere shorten. And as the individual sarcomere shorten, the Z discs get closer together. And if they overlap, as you get these closer together, the proteins don't change in shape or size. They just overlap. So there's a limit to which they can stretch, and there's a limit to which they can contract. And you can actually see that. Right? You can only contract your bicep to a certain point, and you can only extend it to a certain point, and then it actually stops. Right? And you have muscle spindle input telling you, okay, that's about good, right? Somebody grabs your arm and you're like, okay, right? So you have that feedback that's telling you that they shorten, but they only shorten to a certain extent. So the thick and thin filaments don't change shape or length. The overlap changes. And there are linking proteins. So this is important. The dystrophin and other linking proteins, they physically take that inside contractile force and translate it to the outside environment. Because if you're pulling on something and you're not attached to something, nothing happens. So you can have the most powerful vehicle on the planet. But if the wheels aren't grabbing because you're sitting on a sheet of ice, nothing happens. you have to be physically attached to the outside environment in order to translate force. Right? If you've driven at all in Flagstaff in the winter, you might have seen that happen. So these linking proteins transfer the force, pull, to the extracellular tissue. Okay? This is where we'll pick up on Wednesday. Don't forget the homework. Do not forget the homework.